In this example, we did a Google search for acetaminophen as a toxicity reference compound and has identified a link to the ToxBank wiki site. Here is information on all the gold compounds. You can see on the left there's a listing of the hepatotoxins. Here is some data, in vivo data, PK/ADME data, omics, IC50 data, physical properties, and recommended products and sources. So here we provide general information on acetaminophen. What we can do is go to the ToxBank data warehouse, which contains all protocols and data generated as part of the SURAT1 project. And here I'm going to log in. And as a start, we can look to see what investigations data, what protocols have been uploaded. Here are the investigations and protocols describing those investigations uploaded and the current status of that upload. Here's the protocols that have been uploaded. So it looks like there's a variety of omics and in vitro screening data available. So as the next step, let's search for acetaminophen and see what sort of information is available. And here I'm just going to do a structure search, paste it in the smiles. With acetaminophen, we have the information on the compound, a link to the Cosmos database, which contains in vivo toxicity information. There's also information on the hepatotoxicity screening using HEPA-RG cells, proteomics experiment. There's transcriptomics on the reference compounds. There's human skin-derived stem cell data. We can look, for example, at how this study was performed. Let's just look in the dashboard. So if we look at this study, it was not only performed on acetaminophen but on a number of compounds. It's obviously a dose-response study using fluorescent imaging and measuring cytotoxicity, but there is a detailed protocol which we could access to get more information about how the study was performed. Here's some general information, but if we click on the download link, we can open up the complete protocol. So going back to this dashboard view, if we wanted to actually go ahead and download all the data, clicking on the download link for this investigation, we can see the raw data files which can be downloaded. In the results, we also saw that there was a transcriptomics data set. So here is an example. We look at this transcriptomics data set. And we can see again, we can download the investigation. So the ToxBank warehouses uses the ISATAB Universal Data Exchange format. So if we go ahead and save that, this is a document containing all the information about how the investigation was performed. And what we can do is there's a tool called ISACreator, which we can then open this study up, and here we can see precisely the sample information on the sample, the characteristics, the tissues, the cells, and then also information on the doses and controls and units. So all the information about the experimental design, how it was performed, as well as how the processing of this data was performed. As a next step, we might want to download and use the information. So here we can see there's a derived data file. So if I just go ahead and save this target, What that's going to allow us to do is to open up that information that we just saved. This is a tool called Incremap, and what it's going to allow us to do is to look at pathways. So here, because we've used a standardized information, we're going to load, for example, fold changes from the genes, and we're going to load p-values, and this is for acetaminophen, and here you can see it's at 24 hours. So let's go ahead and name those columns. So we've loaded p-changes, p-values, we've loaded fold changes for acetaminophen at 24 hours versus the control. And what this tool will allow us then to do is to do things such as pathway enrichment. So here we're going to look for fold changes greater than 1.5, as well as p-values that are less than 0.01. Here is some pathways that have been enriched, and if we want to see precisely how those genes have been changed on these pathways, we can then look at these differences with red showing higher and blue showing lower.